Hi, this is Miss Litton, and this is 7th period review for the essays and basically the unit for AP Bio. Say hi.
Hi.
All right, what joy do they have? Okay, first thing on homeostasis is going to be any kind of questions about positive and negative feedback and being able to give a strong example of each. Being able to differentiate between the two. In your mind, when you think about negative feedback, does that mean it's like a bad thing because it's negative, like you're wrong? No. It's keeping it at a what? Status quo, right? Status quo. For instance, blood sugar level, right? You want to keep it at a certain percentage that's healthy for your body. If your blood sugar is too high, let's bring it down. If your blood sugar is too low, let's bring it up. Negative feedback, we're bringing you back to the correct level that it should be at in order to maintain homeostasis. Look through your book and come up with several different examples of negative feedback. Most of homeostasis that we think about in our body is negative feedback, keeping standard quo. Positive feedback is amping it up. Positive feedback is where the response makes you have more of a reaction to it, and even more and even more. Usually you're trying to get to some final climactic event. Maybe having the right hormone level so you can ovulate. But the classic one most people talk about is giving what? Birth, right? So the process itself, oxytocin, released from your pituitary gland, makes your uterus contract even more and push even harder against your cervix, which in turn makes it even more oxytocin get released. So it's that repetitive cycle. So I want to say for essays about that, be able to recognize the difference between those. If I give you a scenario, you can go, that is negative feedback, and be able to give me data to support why it's negative feedback, or that is positive feedback, and give me data to support that. You good on that one? Okay. When you get into the circulatory system, structures, and functions, always, for all your systems, think about, hey, this is an adaptation that's super helpful for this particular system. Can you think in the circulatory system of different adaptations that make it helpful? Certain structures. Valves. It's a great one. Thank you. Valves are helpful. Where do you find valves?
Veins.
Why do you need valves in veins? Yeah, to prevent backflow. Sure, that's one reason. What else? Keeps the blood going one direction. What else makes that work? Because veins wouldn't work if they were structured like arteries. Muscles are in arteries, right? But what is it that veins have? They're very what? Thin walled, right? Oh, I know what you're talking about. Now I got you. The muscles contract and push on the vein walls, right? And then that pushing, that compression, forces blood to go in one direction due to the valves. So you want to give me the whole picture. Sometimes some of you just stop a little bit short and you'll say muscles. And I don't know that you're talking about muscles like I was confused just now, and you're referring to arteries, or you're using muscles, and finish the end of the statement for me. Make sure to finish the statement and say, muscles contract and shorten. They push against the vein walls. This forces the fluid in the veins to move, and they can only move in one direction due to the valve. See, that's the complete thought. Claim, evidence, reason. Support it. Give me the reason why that works. When you think about the circulatory system, you want to think about the structures of the circulatory system and why those work together. You want to think about adaptations you can see over the course of a circulatory system. You want to know the parts of the heart and where different fluid goes. You want to know like key things that happen like at the capillary bed, and how we can have something happen like exchange of oxygen and nutrients and CO2 and waste. Talking about those combating pressures, right? Hydrostatic pressure at the arterial end, forcing fluids out. Osmotic pressure is continuous throughout the capillary bed, but it is greater than the hydrostatic pressure once you reach the venual end. Things like that, explaining how our system works. What keeps the blood going? The heart. There's 4 chambers. Why are 4 chambers important? 2 of them what? Collect blood, and 2 of them, why not just have a single pump that's really powerful? Because we're trying to prevent the what of blood? Mixing of blood, because we want to be very efficient. Send the deoxygenated blood to the lungs, coming out of our right ventricle, right? And heading over to the lungs, come back, collect that good blood in the left atrium, send it down in the left ventricle, and we want the left ventricle to pump blood to our systemic circuit. We have adaptations out in those circuits. One I would think of is like the hepatic portal vein. Why is that such a great adaptation?
It bypasses the
It does you're right that it bypasses the pulmonary system for the short term, but that's not what makes it great. What makes it great? Yeah.
It regulates the nutrients in the blood. It goes
Right.
to the liver first.
Right. It doesn't go, it goes immediately to the liver after leaving the capillaries around the small intestine. Now, is the blood always rich in nutrients when it leaves the small intestine? No. It could be nutrient poor, but that's when the liver has an opportunity to put nutrients into the blood. So that's why it's a great adaptation. We know, because we understand how pH affects respiration rate, we understand how osmotic potential and osmotic pressures could influence what happens at the capillaries, right, and could influence whether that exchange takes place. We know that too many nutrients in our blood could raise our blood pressure, right, because it becomes very hypertonic. So we know it's critical to help us maintain homeostasis, that where we have one location where we add something tremendously to our blood, we better make sure we regulate it. So it's great that it goes to the liver to get that regulation. Okay? I can go over any parts of that. I just kind of hitting some big points first out here. Lymphatic system and immunity. You want to make sure, I put innate/inflammatory response because you get the inflammatory response from your innate, what you're born with. You can always have inflammation. But if you want to actually win the battle over whatever's causing the inflammation, that's when you need to have your specialized acquired immunity, right? So for your lymphatic system, that's one of your known systems. So I expect you to know that in great detail. Okay, that's one of the mandated systems. So things like explaining to me the lymphatic system and its main functions, right? To pick up excess tissue fluid at the capillaries, which would look like an epic fail, because that means hydrostatic and osmotic pressure wasn't equal, you had more pushed out than reacquired, so you think that's really bad, that's going to cause edema and swelling. But no, the lymphatic system uses that unequal pressure to capture that fluid and return it, but before it returns it, it passes it through a lymph node, so it works out well, okay? So it collects excess tissue fluid. What else does it do? It produces lymphocytes, B and T lymphocytes, your special forces. Remember, natural killer cells are also, should have been T cells that went to the thymus, but they dropped out. So those cells are still considered lymphocytes because they come from the same lymphoid stem cell. They don't come from a myeloid stem cell that produces red blood cells and all the other white blood cells. I'm talking about granulocytes and macrophages and all those kind of things, right? So it is still considered as coming from categorized as a lymphatic cell. It just fails because it doesn't have any memory. You want to be able to talk about the general police force and the inflammatory response and your players, right? Who's usually first on the scene? Neutrophils, one of your granulocytes, right? That's what you have the most of. What is it doing? Eating. Good. Who might show up a little bit slower but more effective? Macrophages. But before, in order to get out of the capillary, what were they? Monocytes. And they developed into macrophages. And their job is to eat and what? Present. And then you learned a little bit more later that the reason when they present, they're actually presenting on an MHC2, I will help you. So that we know later it's going to kick into this specific immune response, how those two overlap, right? Who else comes up and starts cleaning up around there besides dendritic cells? Go up, and they are going to collect and present as well. Macrophages and dendritic cells also are releasing chemical signals out to the body saying alarm, alarm, alarm. Right? These are all of our complement proteins. They fall into different categories. One is to pump the immune system up. Who is that? Interleukin. That's super stupid. Star Wars Luke, he had to raise the inner, within him. Why interleukin? I know it's bad, but that's what works for me. Interferon, I'm trying to interfere with the viral life cycle, okay, by preventing any more infections. Interferon says guard your doors and windows. cells, be careful so that a virus does not come in. Okay, then we have complement proteins. What do? They drill what? Holes, right? CD4, classic 2A, B, 3C, right? Remember those guys? So they drill holes, right? So those are all proteins, chemical messengers that can enact behaviors, right? They can trigger a cell, they can trigger a signal transduction response. There's another cell involved in the inflammatory response which really does the inflaming. What cell is that? Mast cells. Okay? We learn later that an antibody has to embed the foot of the antibody into the mast cell, and that mast cell, remember IgE, that particular antibody? Okay? It has to be embedded in, and then it has to be attached to an antigen in order to trigger the mast cell to release histamine. Remember, histamine causes vasodilation. It creates greater space between the endothelium cells of the capillary bed. And there's advantages to that. Some disadvantages, but there's some advantages, right? What's an advantage to that? Yes?
Phagocytes can get through more.
Phagocytes can get through more readily. It's more space between them, and they're trying to squeeze out. What is another advantage? Yes?
It increases the heat.
Increases the heat. Might kill out the bacteria. Great. Another advantage. What's another one? It does cause swelling, which is pain, which you kind of don't like, but what does pain tell you? Yeah, something's wrong. You've got a problem here. Okay, so it does do that. Histamines can be detrimental when we overreact to a response like pollen, but more specifically bee stings and things like peanut butter and things like that, where you go into anaphylactic. So much swelling, you're closing off your trachea so you can't breathe, and those types of things. That's an immune system that's just overreacting to the problem. Okay? All right. Good, good. Neutrophils, eosinophils eat the worm. Basophils, another granulocyte in the blood, does the same thing that mast cells do. Complement proteins, just like I'm running through them all in my head. Yeah, we got them all, okay? So that's your inflammatory response. And that is what everybody is born with. That's your Lady Gaga, right? Born this way. Everybody has that response. Then when we talk about our acquired, okay, immunity, you could write a whole essay just on inflammatory response. You could write a whole essay on B cells. You could write a whole essay on T cells. If I want to talk to you about the B cells, what kind of immunity is that? What do they call it? It is acquired. Both B and T are acquired. Humoral or antibody mediated? Humoral or antibody mediated? Humoral comes from your humors, one of the whatever humors in your body, some English, right, reference, and the fluids of your body. It's old. Okay, that's where that comes from. Humoral and antibody mediated. Remember, this would be if we had bad guys, robbers, running around a park, and they're in the streets and the alleyways, right? That's humoral. What would be, take that as an analogy, what would our T cells take care of? If they'd gotten into like a house or a building, right? Two different ways to handle the problem. So your antibody-mediated immunity, how does that get initiated? It always starts with a virgin, okay? So in this case, it's a virgin B cell. And the virgin B cell has what? Receptors. For what? Antigens. Where are these antigens located? On the surface of what? Virus or bacteria, right? It's direct content for the B cells. Direct contact. And that's good because that's where they work too. That's the medium in which they work, right? Out in your plasma. So they come in contact with that antigen and that virgin B cell. But that virgin B cell cannot be triggered unless who agrees? The virgin T cell. The virgin T cell comes in contact with the antigen via what? An antigen-presenting cell, right? Virgin T cells can only be elicited by an antigen-presenting cell, like a dendritic cell, right? Presenting that antigen. Okay. So it's the virgin T cell agrees. Now, what's next? I've come in contact. I'm a virgin B. I've come in contact with the antigen directly. A T cell agrees with me. What's next? Time to make my what? No, not yet. I've got to make the cells that make the antibodies. Plasma cells and memory cells. You are making a clone army, clones, clonal selection of that particular B cell. It's kind of like you have a million virgins sitting in a line, right? Those are all your B cells, a million virgins. One gets tapped, that one steps forward and then makes a million of just that cell, right? To fight off that particular pathogen. So those plasma cells are going to secrete antibodies. If you're going to talk about antibody-mediated immunity, you better tell me a little bit about antibodies, right? We have 2 long chains, 2 short chains. The end of the chains are which are the parts that are variable and specific to that antigen. The base of the Y determines how it's handled. If you want to support it with knowing some of the 5 different ways that, right? Remember I gave you the 5 different immunoglobulin A found in the throat. I'm not asking you to memorize that, but just give me one example of how the base varies the action, right? IgG can cause macrophages eat you. IgE is the one that gets into a mast cell. So, you can come up with 2 in your set, right? Give me 2, so I know that you know the base determines what happens. You're going to say those antibodies are released and can move through the blood, and in turn they can move into the tissue fluid because they can get pushed out due to hydrostatic pressure. They can get pushed out. So those antibodies are moving through your body. Those antibodies will tag and flag that foreign pathogen, that virus, that bacteria for destruction. Now, besides plasma cells, what are the other cells you make? Memory cells. The memory cells are for the future. Yeah. They don't have to be conferred upon by a T cell to say, okay, now you can go ahead and reproduce. They're easier to trigger. When the infection is done, what are those? We had selected 1 virgin, brought her forward, made a million of her. What happens to her clone army? Apoptosis, except for the memory cells, and they stay on, right? So that's antibody-mediated immunity, taking care of the free-floating viruses and pathogens. Now, let's talk about cell-mediated. Let's talk about cell-to-cell combat, about T cells. Again, it always starts with a what? A virgin. Starts with a virgin T cell. Okay? That virgin T cell has to be stimulated. Oh gosh. I wish I could erase that. Okay. What is, I was going to say, okay, let me try some other words. What could start a signal transduction pathway in that T cell? Antigen presenting cell. Okay? An antigen presenting cell with MHC2, I will help you. Okay? MHC2 presents the antigen to the T cell. And this is stabilized by a CD4. I'm for you, not against you. Right? And this will trigger that virgin T cell to grow and produce 3 types of cells. Okay, what are the 3 types of cells it will generate?
Cytotoxic, helper, and memory.
Perfect. Who coordinates? Who is the general? Who says, let's do this? Helper T cell. It's releasing lymphokines. It's saying, hey, let's fight this. It's going to produce, your interleukins and say, let's fight. Coordinate the entire response. Okay, those are your helper T cells. Cytotoxic T cells, what do they like to do? Kill. How do they know who to kill?
Their mark.
How the antigen is presented. Okay? It does not know, right? It can't see the cell. So it can only tell by the MHC the antigen is getting presented on. If the antigen is presented on an MHC, then you're done, right? The T cell will stabilize that binding with CD8. Exactly. And so then it knows that's the one I need to kill. And so it'll kill anyone who displays a foreign antigen on an MHC1. Okay, and then we also have what kind of cells? And those are for, more easily stimulated. Okay, and then those cells all commit suicide. All right? Now, problems with that, I would probably bring up things like anaphylactic shock, autoimmune diseases. Right? Okay. All right. Adaptations of the respiratory and the digestive system. We talked about some adaptations of the circulatory system. Let's talk about some adaptations of the digestive system. Yes.
The epiglottis?
Sure. But that would be of the respiratory system, because that's what it's really protecting.
But like it kind of like makes sure the food doesn't like choke you to that.
True, but that's more of a respiratory system adaptation.
Oh wait, well, what about the pepsin thing?
Okay, I've heard a couple things. You know what one I would lead with? Microvilli of the small intestines. I would have led with that. Okay, it's your best adaptation you got out there. Okay, why? Right, for what? Nutrient absorption. Okay, those are the kind of things I would lead with. That's a really good adaptation, right? What are some other adaptations? Okay, how else could we say that? I agree with you. Different types of mechanical digestion, right? So we could talk about the teeth that you have, for whether they're broad or flat, or you have ones for cutting and tearing, or you're a bird and you use rocks to break things up, right? Those are all different adaptations for mechanical digestion. Let me just tell you what I've read so you can have a little insight into the patheticness that I've been exposed to by some of you. I would say, please give me some adaptations about digestion. Some teeth are sharp for eating meat. Adaptation number 2, some teeth are broad. Adaptation number 3, you laugh. I've read a lot of your essays, okay? Some use rocks. What is that all about? Teeth and mechanical digestion, all right? So be wise, okay? Be wise, because I start to get annoyed. Okay, what are some other adaptations of the digestive system? Yes?
Does pepsin and pepsinogen, like how it activates?
Okay, the ability to store what? Inactive precursors as zymogens, so that you do not digest your own body tissues. I'd say that's a great adaptation. What are some other adaptations?
The length of your digestive tract.
The length of your digestive tract, depending on the things that you eat. How about that you can start digestion in one organ and finish it in another organ? So it has time to be worked on, right? We give some enzymes in our mouth where we can focus on carbohydrate digestion. We can change the pH in order to create the right environment for our particular enzymes that we're working on. Protective things like mucus, or the pancreas being able to secrete a bunch of bicarbonate to handle the acidic environment coming out of the stomach. We have some redundancy, right? We have pancreatic amylase and we have salivary amylase, in case we don't have one or the other. We have some redundancy there. Okay, so those are all adaptations that you could talk about. We could talk about specialized area for reabsorption of water, because water is critical for maintaining homeostasis. All right, and then we can talk about the specifics of digestion in just a minute. Let's talk about some adaptations of the respiratory system. Here you go.
Epiglottis?
There we go. To ensure that food goes down the digestive tract and not our respiratory tract. What are some other adaptations?
The nose and all that does for air.
Yeah. Different, like we have ways of filtering the air, right? We have hair, right? We have moisture, and we have, but you know what I would lead with?
Alveoli?
Yes, I would lead with alveoli and the microscopic alveoli, because it increases
Surface area.
for absorption and for exchange of all of our gases. I would have led with that, okay? Anything like that I would have led with. What else are some adaptations that we have in our respiratory system? We have a trachea, it's permanently open, right, using cartilaginous rings. What else do we have? Yes?
Cilia.
We have cilia in our trachea, and that is going to help clean things out of our lungs, right? Anything else?
I don't know what birds.
Oh, it's a great adaptation. Birds have a posterior sac, lungs, and an anterior sac. It's a great adaptation so that the air moves unidirectionally through the bird's respiratory tract. That's great. Counter current, which makes me think about fish and gills and the counter. So you could just talk about counter current and how efficient that is, and you could reference birds and you could reference gills, right? Yeah. So think big picture, think structure and function, and how that helps an organism maintain homeostasis. You could talk about hemoglobin, right? You could talk about that in the circulatory system. That's one that really you could use in respiratory or circulatory or respiratory system. The transport of blood gases, right? Hemoglobin. All done, sweetheart?
Yeah.
Questions? Okay. Hemoglobin acts differently considering what pH is in. Who would know that blood pH changes, right? So, and the temperature. So it facilitates the uptake of oxygen and the saturation of hemoglobin in our lungs and the release of oxygen out at our tissues where the pH is lower. Why is the pH lower out in our tissues? Because why?
Hydrogen is like broken down. It's broken down to hydrogen.
Yeah, okay, so CO2 will combine with water and form what?
Bicarbonate?
Carbonic acid, right? And you use carbonic anhydrase to do that. Then carbonic acid will break down into bicarbonate and hydrogen ions. The hydrogen ions from that, from carbonic acid becoming bicarbonate, lowers the pH, which then makes hemoglobin less likely to hang on to oxygen. Okay? And then it will release it. Then CO2 can travel in your blood safely as bicarbonate, dissolved into the blood. And then hydrogen will hitch a ride on hemoglobin. Now you're back at the lungs. And we have to talk about partial pressures of gases. Like we kind of left that over there. We always go from a higher concentration to a lower. Right. So in your lungs, you have a higher concentration of oxygen. It's going to move from a higher concentration to a lower concentration in your red blood cells, kick off the hydrogen. Now the hydrogen is available to connect with who? The bicarbonate, right? And then it will form carbonic acid. And then carbonic anhydrase will break down carbonic acid into CO2 and water. And then the CO2 can be exhaled. Make sure you're clear on what happens with gas exchange at both the lungs and the steps that takes place there. We went over it multiple times. And out at your tissues and the steps that take place there. Be very comfortable with those reactions. Okay, the other thing you have to be comfortable with is your highly suggested reading and thinking. Right? Make sure that you're good with that you go over those, you don't ignore those, but that you could respond to those types of questions. I'm thinking through your tests, all of your tests. That's what I'm doing in my head.
Do they look bad? That
No,
looks like
I
a yes.
think we've talked about pretty much everything. Yes.
So because the test is on Friday and that's
Yes.
rally day and also you're not here, how would
Right.
it look different from a regular test?
It's going to look like you're going to get, each class has a different test, like multiple choice portion of the test. You're going to get a single sheet of paper, and you're going to write your answers on that single sheet of paper.
Oh, we did it once.
Oh,
Like
like we did for the chapter.
Yeah, you won't have like a, like a Scantron. You'll just do it all at once. You have a place for you to put your Scantron. What you would put on a Scantron, you'll just write your answers down on it, and then you'll write your essays on that paper. Okay? You're probably going to want some scratch paper, so be prepared to cough up a piece of notebook paper tomorrow in class, and then we can staple that to your sheet so you have scratch paper to kind of jot things on. Does that make sense? So have one of those for me, and then I'll staple it to your test. What else? Okay, when you look at the questions that I said here, that I gave you on potential essay questions, I said name and describe feedback mechanisms involved in homeostasis and describe how various body systems can contribute to the process. Did we talk about that? Okay. Assume you are a red blood cell and describe your journey through the human body starting from and ending at the right atrium. Describe any changes you notice in blood as you travel through the organs and tissues of the body. Be sure to focus on what happens physiologically at these areas: the muscles, the lungs, the kidneys, the intestines, and the liver. What's going to happen at the kidneys that I haven't taught you yet, but you know what happens at the kidneys? It's getting filtered of waste, right? All right. And we talked about the other ones. This graph below shows the response of the human immune system to exposure to an antigen. Use this graph to answer parts A and B of this. So question A says, describe the events that occur at period 1 as the specific immune system responds to the initial exposure. So here at day 0, you've been exposed initially to the antigen. This is the second time. This is like you get a polio vaccine, and then you come in contact with real polio. And what does this tell you? This says antibody titer. It's measuring how much antibody is in your serum. What is this telling you? The second exposure, what? It's what? You have a lot more, right? A lot more antibodies upon the second exposure. That's really why you get it this first time. Is the second time you can see it responds much quicker and more effectively. Now, does it slowly, gradually degrade over time? Yes, but you still have them in your body. You're not ever going to get rid of those antibodies. All right, and then, oh, this was a mini me question. After school one day you notice that you are starving and decide to go get a cheeseburger. As you are eating, you think back to all that you know about the digestive process. Assuming you have protein, meat, carbohydrates in your bun, and fat, the meat, the mayo, and the cheese, describe how each nutrient is processed, digested, and absorbed as it passes from the mouth to the anus in your complete digestive tract. Okay, let's do it. Let's start in the mouth. Let's do each of them individually. Let's start with the meat. Okay, what's going to happen to the meat in the mouth?
Mechanical.
Good. Is that it? Using what? You're going to tell me mechanical digestion using your teeth. It's then going to go down your. Esophagus. Esophagus. I know now you're only going to be able to say it like that. Your esophagus. What's going to move it through my esophagus? What's it called?
Peristalsis.
Peristalsis. It's then going to go to my, what's going to happen in my stomach?
Mechanical digestion.
Always lead with mechanical if it's there. Okay, so mechanical using muscles. Chemical using inactive precursor, which was inactivated by or activated by
pH level.
lower pH level. Good. What's protecting my stomach walls? Mucus. Secreted by the worst kind of pie ever. Pyloric gland. Pyloric mucus pie. Did I not tell you that in class?
Oh, yeah.
You're like, I blocked it. Okay. So you leave the stomach and now you're moving into the small intestines. You still have protein. Is the liver doing anything for us?
Secreting
Bile. But we're going to deal with fats later. It's not really affecting protein digestion, right? Just the fat that's in the protein. All right. What else? Pancreas do anything for us?
Yes.
Yes. Trypsin and chymotrypsin, right? Those are both digesting proteins for us, and then the small intestines with peptidases, or just you can just say completes digestion, and then you have absorption in the small intestine. Okay, meat done. What's next? Bun. Mouth.
Chemical.
Start with mechanical teeth. If you do it in the same way every time, you're less likely to make a mistake. So mechanical teeth, mouth. What else? Salivary amylase. Start breaking down carbohydrates. Again, esophagus, stomach. Anything happening there? Mechanical using muscles. Small intestine. Liver do anything for us? No. Pancreas do anything for us? Pancreatic amylase. Small intestines do anything for us? Maltase, sucrase, right? Finishing the digestion, and then you have absorption. Finish and absorb. The only one it doesn't finish is fat. But it packages it. But it doesn't finish it. Okay? Let's do the next one. Let's do fat. Mouth. Mechanical teeth. Esophagus. Mechanical muscles. Small intestine. Liver do anything for us? Yes. Liver makes bile, stores it in the, secretes it into the duodenum or beginning of your small intestine. What's that word she just said? In the small intestine, the beginning of it is called the duodenum, 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 whatever you want to call it. And what does bile do for you? I was a twin today, by the way.
It surrounds the fat.
It emulsifies fat. That's a word you're going to want to know. Emulsification. Okay? It emulsifies fat by breaking that fat into smaller pieces, right? Did I remember to hit record? Yeah, I did. Okay, so you with me? Now, if when you hear this, you're like, I don't process it with her just talking at me. It's too much. I don't understand it. This is hard. Did I give you a chart with all of this? Yes. Okay. So it emulsifies the fat. Does the pancreas do anything for us? Yes, it secretes lipase, which breaks down the fat, into glycerol and fatty acids. Then the key thing about these is that you have the lacteals, right? Your small intestine is going to help package it and then move it into the lacteal, which is part of your lymphatic system, inside each microvilli. So it'll be transported that way. Significance too is the absorption of water. Small intestines absorb water, but the large intestine, the colon, ascending, transverse, descending, rectum, anus, they're also getting some more water out as well. Okay, and then considering the respiratory tract, we have talked about it, we have talked about it, and we have talked about it. Okay, you're good to go. All right, and remember, if you're in the rally, 4th period, tomorrow, 3rd period, Thursday, I'll remind me, Thursday, 7th period, if you want to have enough time. Okay? Make good choices, have a piece of